In 2025, over 500,000 people will die of cancer in the United States. The cause? A lack of early detection and prevention. But that is soon to change, as researchers have now discovered that cancer DNA can be detected in the blood years before diagnosis.
What? That's amazing. How do they do that?
Researchers at Johns Hopkins studied blood samples collected in the 1980s to 90s from 26 people who were diagnosed with cancer just 6 months later. Researchers were specifically testing the ctDNA.
What is ctDNA?
ctDNA is short for circulating tumor DNA. You see, when you have cancer, your cells become abnormal. They can't stop dividing and multiplying, which eventually leads to a huge clump of these cells. These become what are known as tumors.
I'm confused.
Stay with me here. Let's use a simple example for the entire experiment. Imagine in school, one kid stops listening to the rules and starts running around the classroom. That convinces other kids to do the same, and pretty soon the whole class is out of control. Now imagine one of these kids leaves the group and runs down the hall. That's like a piece of the tumor dying, breaking off, and floating into the bloodstream. That's the circulating tumor DNA, or ctDNA. Then, researchers tested these pieces of tumor DNA for mutations.
What do mutations have anything to do with this?
Think of DNA like a set of instructions for how a cell should behave, kind of like a rule book. A mutation is like someone scribbled on the rule book with a marker or started randomly erasing. Suddenly, the cell thinks it's supposed to keep dividing nonstop.
So it's like the kid thinks running around the room is the new classroom rule.
Exactly. The mutation rewrites the rules and the cell goes rogue. That's how cancer starts. Researchers check the DNA in the bloodstream to see if any of these rule-breaking instructions, those mutations, are floating around.
Oh, okay. So when researchers are checking for mutation, it's kind of like teacher asking why the kid is behaving that way.
Yup, researchers performed two different tests. One's like a full body scan, and the other's like a spotlight on known trouble areas. The whole genome sequencing looks at the entire DNA. It's like reading every page of an instruction manual to find typos. The targeted gene panel skips the specific chapters we already know are linked to cancer, like looking at a list of usual suspects.
So one is like searching the entire library and the other is checking just the mystery section.
Exactly. And by doing both, researchers could catch both well-known and unexpected mutations that lead to cancer.
That makes a ton of sense. But what's this thing about knowing years before?
Researchers had two different types of blood samples, also known as plasma, that they tested. One was from 6 months before, like I talked about previously, and one was from 3 years ago, before the cancer diagnosis.
Well, did it work?
Yep, it actually worked very well. In one sample, 98% of the mutations detected in the 6 months blood sample was also found in the one 3 years before. And for another sample, 80% of the mutations were also detected. Also, these mutations detected in the ctDNA match the cancer that the patients were later diagnosed with.
This is revolutionary. If we can detect cancer years before, doctors can prevent the cancer from growing, and the earlier you catch the cancer, the more treatment plans are available.
It is very revolutionary. This study demonstrated how early ctDNA can be detected by early cancer detection tests. However, it is in a very early stage, and right now the test only catches cancer about 31% of the time when it's 6 months before diagnosis. To detect cancers 3 years or more prior to diagnosis will require a lot more higher sensitivity than detecting cancer that's 6 months. It'll probably be years until this method starts to be used in hospitals. But in the meantime, it sets a great foundation and benchmark for other researchers to build on and create technologies that can test ctDNA in blood more efficiently.
Holy yap! That was a lot of information, but I learned a lot.
Don't forget to like, subscribe, and turn on notifications. We hope you enjoyed this video.